protocol offers a Biotin-labeled platform for the study of protein-nucleic acid interactions that proves to be robust, reliable, efficient, and affordable. The same batch of the Biotin-labeled nucleic acids can be used over a long period of time, maintaining reproducibility of experiments. Finally, all Biotin-labeled assays described here can be performed within a day and do not require special equipment. Demonstrating the procedure will be Lina Yu, a postgraduate from our laboratory.
Prepare MyoB and Move10 constructs and transform them into the appropriate bacteria. To extract MyoB, pellet the cells via centrifugation and resuspend them in 20 milliliters of ice-cold Dolbeck's phosphate-buffered saline, or DPBS, buffer. Sonicate the bacterial suspension on ice. Then, centrifuge the lysate at 12,000 times G and 4 degrees Celsius for 15 minutes, and transfer the supernatant to a fresh tube. Pre-wash glutathione Sephiroth beads and incubate them with the lysate at 4 degrees Celsius for 2 hours. Centrifuge the mixture at 750 times G and 4 degrees Celsius for 1 minute to pellet the beads. Rinse the beads with 10 milliliters of ice-cold PBS 8 times. Then, add 1 milliliter of elution buffer, incubate the beads at 4 degrees Celsius for 10 minutes, and elute the beads via centrifugation. Repeat the elution 6 times and pull together the 6 fractions. Transfer the eluted proteins into a centrifuge.